So cells are usually, well, they are too small to see with the naked eye, so we have to use something called a microscope. And here we have an image of a microscope in this video. So visible light passes through a suitable specimen, for example, say an onion cell, and a series of lenses magnify an image that is formed, which you can then view. This particular microscope we see before us is called a light microscope, and that can give a useful magnification of around 400 times, which means that the image is about 400 times larger than the object that's being observed. The contrast between different structures in an image can be improved using some chemicals known as dyes or stains. For example, the nucleus of an animal cell can show up particularly well if it is stained with a dye called methylene blue, and plant cells often show up better when stained with iodine solution because they contain starch. A typical animal cell is about 1/14 of a millimeter in diameter. Now this is of course a rather clumsy term, because what does it actually mean in terms of 1/14 of a millimeter? So we as biologists use smaller units. For example, 1 meter contains about 1,000 millimeters, and 1 millimeter contains about 1,000 micrometers. So a typical animal cell is about 25 micrometers in diameter. So now what we're going to do is go through the parts of the light microscope which you will be using in class. So we'll start off first with the eyepiece which you observe the specimen through. There are sometimes 2 of these or 1 of them, and you should be able to focus them independently of each other if there is 2. The next part of the microscope is the eyepiece focus. This allows the eyepiece to be focused independently of the other one, which compensates for the differences we have in human eyes. The next one area of the microscope we're looking at is the objective lenses, and there are usually 3 objective lenses, in rare cases sometimes 4, and they can be moved into position for viewing. Each will have a different magnification, usually 2 times, 10 times, or 40 times. The next part of the microscope is known as the stage. This is where you place your microscope slide which contains the sample you want to observe. The microscope slide itself is a rectangular shaped piece of glass that is covered with a cover slip, and between the cover slip and this piece of glass is the specimen you are going to observe. The next part of the microscope is known as the stage clip, and this holds the slide into place, preventing it from moving around when you're moving your objective lenses. So the stage controls are used that allow either coarse movement or very large movements of the stage to move up and down, or a fine control, which means that you're controlling it in a very fine way, so there's small movements for every turn of this control. Okay, so it moves the stage either up and down or left or right, allowing you to manipulate the position of your specimen to allow easier viewing. The lamp control is on the side, and this switches the lamp on, and you can also adjust the brightness of the light source going through the specimen. The next part of the microscope is the light source, and this is usually in the form of a lamp, and it supplies light that shines through and illuminates the specimen from below. However, remember that direct sunlight is never used because this will blind you. The condenser allows the adjustment of the light settings and how much light is getting through to the sample on your slide to allow ease of viewing of your specimen. And the condenser control further underneath moves the lever to control the size of the opening and the amount of light getting through into your sample. We have the focus knobs on the side. The one on the inside is known as the coarse focus knob, and this will allow you to roughly focus the object by again moving up the slide or down the slide stage. And the one on the outside is called the fine focus knob, which means turning that only makes small adjustments to the stage, allowing you to fine tune the focus. And then we're back to the eyepiece. So those are the major parts of the microscope which you will be using today, and which you have to become familiar with and learn how to use and manipulate your specimen underneath the microscope.